Hello everyone, my name is Mariam Sulaiman and I'm really excited to share my first experience with NF4 and Nextflow. This project started out with something very simple. I had a set of FASTQ file which I needed to derive meaningful insight from. But what I didn't know was how much I would learn about reproducibility, automation, and troubleshooting. Here is a quick overview of my journey, how I explored Nextflow, picked up NF4 viralrecon, ran my first reproducible pipeline, overcame early beginner struggles, and finally detected the Kappa variant. So let's start from the foundation. What exactly is Nextflow? Nextflow is a workflow manager that helps to handle all the behind the scene chaos of running bioinformatics pipeline. It is portable and scalable, and it helps to automate complex bioinformatics analysis. Basically, you tell Nextflow what to do, and it figures out how to do it efficiently and consistently. It uses containers like Docker and Singularity for reproducibility, and these containers have pre-installed libraries and packages. It is written in its own domain-specific language to run the processes, and this enables modular and reusable pipelines. Also, it is compatible with cloud and local systems. So why exactly do workflow management matter? For me, when I started out with manual analysis, I realized quickly that it wasn't entirely sustainable for me because I had to run multiple tools and scripts, which was error prone. But with workflow approach, a standardized pipeline gets me the results I need. Also, manual analyses are very difficult to reproduce because you sometimes lose track of the steps you take. But with workflow, everything is fully documented and reproducible. Manual analyses are also very prone to version conflict, but workflow are containerized for consistency. And manual analyses are very time consuming too, but with workflow, you have a parallelized and efficient analysis. So as someone who was just starting out, this structure of the workflow was a lifesaver for me. So I chose NF4 viralrecon to run my analysis based on two main reasons. The first being that it is specifically built for viral genomes, and this fits perfectly with my SARS-CoV-2 dataset. Also, it is community curated, that is, it has been tested, reviewed, and standardized by a group of experts. And with one command, I was able to run all of this process, starting with my FASTQ file. I moved to the quality control, the alignment, variant calling, and eventually generated my reports. What I like about this analysis was that it is built with tools that are recognized, like the FastQC, the Hiver, MultiQC, the Bowtie 2. Also, the whole process was fully transparent. It delivers organized reports and reproducible outputs so that whenever you need to run that same analysis, you get the same results at each point in time. And also, it is a perfect entry point for learning reproducible workflow. To put NF4 viralrecon to test, I retrieved 5 SARS-CoV-2 Illumina samples from the NCBI SRA, and to my surprise, in less than 30 minutes, the whole process was completed with everything fully reproducible with Docker. And one key insight I derived from this analysis was the detection of the Kappa variant. One of the 5 samples with the ID ER5743893 turned out to be the Kappa variant with 2 key mutations, which are the E484Q and the L452R. And this is what running the pipeline looks like. This command kicked off the entire process for me. All I had to do was just to specify the part of my inputs and the output directory. And this is what my input file looks like. The sample sheet containing the paired head reads. And with just this command, Nextflow helped me orchestrate the tasks, called every module, tracked the log, and managed the dependencies while keeping everything fully reproducible. All I had to do was just to click enter and wait for my results. And seeing this interface of my analysis completing successfully was such a big win for me because I got to see that my samples passed the QC and there were no missing files, there were no errors, it was just a clear run, all within 26 minutes. To further validate the results I got here, I explored other tools. Using Integrative Genomics Viewer, I aligned my reads to the reference genome and visualized the coverage depth and the mutations. Also, using Galaxy, I filtered the SNPs to the high confidence ones and finally generated my variance table. Using USHA, I performed lineage assignment, which was what helped me track my variant as the Kappa variant, highlighting the two spike mutations. So this was where everything came together for me, how I moved from the raw reads to meaningful biological insights. And this all run taught me more than running pipeline. I was able to learn that reproducible workflow helped to simplify complex analysis. Also, containers like Docker make environment setup effortless. And the automation made me focus on my research question, that is the biology, instead of focusing on the configuration or environment setup. And standardization helps to attain faster and transparent results. In the context of global health, reproducibility matters because speed and reliability are critical in genomic surveillance and research. Also, standardization helps to ensure that your results are consistent and comparable. And Nextflow and NF4 empowers researchers globally, regardless of setup, because they are forums where you can actually put out any difficulties you are facing and you get people to respond to you. And my experience shows how accessible tools can accelerate discovery. Although everything wasn't on the top requirements, unclear error messages, part confusion, and the pipeline restarts after every error. Although this was frustrating at first, but along the line, I learned how to read my logs carefully, how to check documentation, how to actually understand error messages to correct what was going wrong. So these struggles were not set back for me, they were learning in motion, and that was what contributed to the results I eventually got. Thank you everyone for listening, and thank you to the NF Core and Nextflow team for putting out tools that empower people to start, learn, and contribute. Here are my GitHub and LinkedIn profile. I would love to continue the conversation. And remember, every successful pipeline run is a step towards a better science.